is going to start in the next weeks, probably now, and how the international standards can help and support to make this mass immunization safer. We have three speakers, three ladies. We will have in sequence. Sorry, give me just a second, and apologies for that. We will have Miriam Sturkenboom first. She's a professor in observational data analysis from UMC Utrecht in the Netherlands. She's also leading the Access, my computer has the screen so busy, Access project, which is funded by the European Medicine Agency to prepare the protocols and infrastructure for the immunization. She's also chairing an association, VAC for Europe. So she will speak the first. She will be followed by Anja van Haren. She works at CBG in Netherlands, second Dutch person for us. She is very much engaged in pharmacovigilance for a long time. Is it at CBG or at the European Medicine Agency? And she's also the UDRA Vigilance Coordinator. And as such, she has contributed to develop international standard, namely ICSR, standing for Individual Case Safety Report. The third speaker is Malin Fladvad. She is a Swedish lady working in Uppsala. She is the product portfolio manager for WHO drug, or WHO drug, that is the medicinal product dictionary maintained by her organization, which has the purpose to work on pharmacovigilance around the globe and also working on alerts. Then we have a panelist, another gentleman, Robert van der Stichele. He's a GP. He says he's retired, but he's not so much retired. She is a professor at the University of Ghent, and he is very much involved also in the UNICOM project in various domains. For us, in Work Package 1, he is engaged to represent IHD and has an in-depth knowledge of pharmaco issues, pharmaco-epidemiology issues, and so forth. So he will add value to the dialogue we are going to have after the presentations. Now, I give the floor to Miriam Sturkenboom. I recommend that our speaker close the camera whilst Miriam is talking, and you will open it again when it will be your turn until the full discussion. Okay, so Miriam, the floor is yours.
Well, thank you very much, Christian, for the opportunity to speak here to this expert audience. And my task today is to set the stage for the discussion today, and that is to talk about how to measure actually the exposure to COVID-19 vaccines, which will be needed for post-introduction monitoring of the novel vaccines. Next slide, please. So this is an overview, today's overview of the COVID-19 vaccine landscape that is ahead of us. A summary capture of the London School tracker, actually, where you can look at this. And as you can see in this graphic, there are a lot of vaccines in development. And actually, in total, there are 274. 58 are in humans, so either in phase 1, 2, or 3 clinical trials. And we have a first vaccine, the Pfizer vaccine, which was just licensed for emergency use in the UK this week. So what is novel about the COVID-19 vaccines is that these vaccines have not been used before, clearly, but also there are many different platforms that are being utilized to deliver the antigen, and some of them are novel and haven't been used for human vaccines before, such as the nucleic acid, the RNA, and the DNA platforms. And as you can see from the graphic, there are actually quite a lot of vaccines that are being built on these platforms. And the first vaccine, the Pfizer vaccine, is also built on an mRNA platform. So this is the novelty. And it's also very exciting because it means that they can be made available much faster. So they're novel platforms. What is important to realize is, whereas there are a lot of trials ongoing, so far there is very little EU trial experience. So most of the trials have actually been conducted outside of the EU. So there's only the Pfizer has been recruiting in Germany, and that's most of the experience we have in Europe. Next slide, please. So here you see an overview of the characteristics of the first COVID-19 vaccines that may be licensed. So currently the BioNTech Pfizer vaccine licensed already in the UK for emergency use and will be coming available also for Europe. I think EMA is counting on mid-December, and also FDA will be reviewing and probably licensing or at least allowing use of the vaccine. Then quickly coming will also be the Moderna vaccine, also built on an mRNA platform, and then subsequently the Oxford AstraZeneca vaccine which is using a different type of platform, a viral vector platform. So you see here the different platforms, the doses. Most of them will require 2 doses. They have been formulated in different ways, have shown to be highly efficacious as far from the interim analysis. And what makes them different and also important difference will be their usability in terms of cold chain requirements. And for example, in the BioNTech Pfizer vaccine will need to be stored at very cold temperatures between -60 to -80, which will pose issues, I think, in the rollout of the vaccine, whereas other vaccines may be refrigerated in a usual manner. So they have different characteristics and will be coming quickly. Next slide, please. So EU has contracted with 6 vaccine manufacturers at the moment. Initially, they had contracts with AstraZeneca, Sanofi, and Johnson & Johnson. But since the data becoming available, they have also been contracting with Pfizer, Moderna, and also with CureVac, which is a German company that they have also acquired doses there. The way the European contract is created or made actually is that there will be a fair distribution across Europe. So initially, so it means that based on the population size, these doses will be distributed equally across European countries. Next slide, please. So because of the speed by which these vaccines are being made available, which has an implication because many of the trials, actually the phase 3 trials are still ongoing, and the current use opportunities are based on interim analysis. So that means that the longest duration at the moment is about 2 to 3 months of follow-up. So there's very little, even if there's very little follow-up currently available, although it will become available at the end. But we will know up till 6 months, and so there is limited knowledge there. As I said before, they're all novel vaccines that have some of them have actually platforms that haven't been used before, and the circumstances in which they need to be provided can be challenging, such as the cold chain or the requirements on keeping them in -70. So if they are defrosted, it may be that actually they lose their effectiveness, and this has many implications, I think, for effectiveness potentially. So it's very important to monitor the benefits and the risks. We all know that it's more important now, I think, than ever to keep the public trust there. so that there is really trust that these vaccines are safe, and that people are not guinea pigs if they use this. And we can see this popping up in the media already. So the Access project has created many template protocols for EMA and to be prepared. So they have been working in the past 6 months to be prepared to do the post-introduction monitoring. And all depends on the ability to establish that we actually can measure which patient received which vaccine. And that's the topic of today. So next slide, please. So very briefly, what does Access do for EMA? First of all, they identified a list of adverse events of special interest, those events that we want to pay special attention to. If they occur, they might have the potential to derail a vaccination campaign, so you want to be prepared. They may not occur, but at least you want to be prepared. And then there's several protocols to actually do European collaboration on the calculation of background rates, safety assessment in various ways, effectiveness assessment, coverage monitoring, and integration of benefit and risk. Key thing learned from the H1N1 pandemic was that there is great potential in Europe to collaborate rather than every country doing its own thing, where you may have bigger scale and an ability to compare between vaccines better if you do cross-country collaboration. So currently, background rates are created in 7 countries, and there is a feasibility assessment ongoing of organizations and governance to be able to implement these studies. At the moment, it's not fully clear who will be paying for the actual implementation of those protocols, but we trust that will clarify soon. Next slide, please. So the question there is whether it's the companies or it will be public funding. So which persons may be targeted? WHO in their SAGE committee have had quite some deliberations already over the past periods, and they came up with a values framework. And you see a picture here, basically looking at different values of who you and why you should vaccinate certain people. And this values framework is being utilized by many of the NITECs in the countries to decide on the rollout strategies in the individual countries. It's important to realize that every country can decide that themselves. So in the Netherlands, for example, they will start with nursing homes and at-risk elderly, but other countries may start, like the UK, to start with the healthcare workers. So there are choices. Although all, I think, will at the end have same rollouts, similar rollout strategies, the sequence may differ. Next slide, please. So we know now that the vaccines are coming. They may have a great success and give back our lives a bit in terms of mobility, but we also know that there are a lot of questions about the safety and that the success will be only there if at least 60 to 70% of the population will be vaccinated. That's what herd immunity would require, is simulated now with modeling. So it will be really important that trust is maintained and therefore that post-introduction monitoring will be effective. And so whereas there are many plans now and protocols ready to implement such monitoring, the key question will be whether at the end, when there will be vaccination, we will be able to see which individual received which vaccines. And on the left hand, you see how COVID vaccines were labeled during the clinical trials. So they had a nice label on the tube. Once they are being rolled out, and on the right-hand side, you see pictures from the H1N1 pandemic rollout, you see these mass campaigns in sports halls or people lining up at GPs on the bottom and being vaccinated. And the question is whether at that moment in time, it will be recorded which vaccine is actually being provided. And that is especially important given the large number of vaccines that are coming up, the fact that you need 2 doses, and that there is not a requirement to do actually labeling or barcoding on the primary package. So there are challenges ahead, I think, and we realize that it's needed that we have this discussion about this. Next slide, please. So, so here are the questions that we have as of to date. The Pfizer vaccine is coming. Not all of the countries in the EU have decided on their rollout strategies. the immunization registers are required by the European Commission. They had a letter in mid-October what they would require. And they require countries to have an immunization register, but it's not clear how data will be entered in that immunization register. And I think that is where many countries currently are discussing and how this will actually be implemented. So, for example, how do you capture that people in nursing homes are being vaccinated, in GP practices, at workplaces, in mass campaigns? And that's, I think, the variability that we may see from one country in the other. So how do we know the details on brand and lot number so that we know if there is an issue with a specific lot because there may be a production error, that not the entire vaccine is implicated or other vaccines as well? Next slide, please. So I think this is what my presentation was. If you have questions, I guess we have them at the end. So yeah, thank you very much for your attention.
Thank you, Miriam. Thank you a lot. Yes, indeed, the questions, you can raise them at any time, as I said, in the Q&A box in the facility. I encourage you to do so. And we will collect these questions and interact with the speaker and the panelists at the end of the three presentations. Thank you very much, Miriam, enlightening for this introduction. And now the floor is for Anja van Haan, and please go. The floor is yours.
Thank you, Miriam, for setting the scene. And let's start with a high-level view of the EMA COVID-19 vaccine safety monitoring plan on the next slide. This document is published on the EMA website, and where Miriam has outlined that there will be studies to monitor the safety. One of the other main pillars is the collection of spontaneous reports of suspected adverse drug reactions. And upon authorization, the vaccines will also be subject to additional monitoring. That's a specific status. And this also aims at enhancing the reporting of the suspected adverse reactions. In this plan, it's also clearly stated, just like Miriam emphasized, that it's really important to have precise information on the vaccine brand and batch number. If we go to the next slide. We have been in a pandemic situation before, about 10 years ago with the H1N1 vaccination as well. And there were some lessons learned. And if you look at the graph from a publication from Xavier Cours from EMA, it shows that the curve of the number of reports of suspected adverse drug reactions received at the EMA, it follows a similar line as the number of individuals being vaccinated. So with this graph in mind, we are preparing for the worst when it comes to the volumes of reports of suspected adverse drug reactions being processed. Also, what we have learned at that time, that the batch number was missing in about half of the reports that was submitted to the EMA. So it's really important that we try to do a better job in collecting the batch numbers. Next slide, please. So let's have a look at the flow of those reports of suspected adverse drug reactions that we also call individual case safety reports. It always starts with a patient consumer who's giving a medicine, or in this particular case, a vaccination, a healthcare professional, and we have national competent authorities, pharmacovigilance centers, and the pharmaceutical industry. So the patient consumer who experiences an event, they can contact the healthcare professional, but they can also contact the competent authority, the pharmacovigilance center, or the pharmaceutical industry. Similarly, the healthcare professional can also contact the pharmacovigilance center to report, or they can contact the pharmaceutical company. Please press, yes, and press again. Just keep pressing. Yeah. So those competent authorities and pharmacovigilance center, all the reports that they receive are forwarded to the EMA, to the ultravigilance database. And from there, the reports are disseminated to the pharmaceutical companies as well. If you press again, you will see that all those reports that the pharmaceutical company received are sent to the EMA and then forwarded to the national competent authorities. So each country is able to have a complete data set of their own country if they want to, and also the pharmaceutical industry will have a complete data set of all the reports that are available. And if we press again, you will see that the EMA will also make all the EU reports available to the WHO. So this is the flow of the European cases. If we go to the next slide, you'll see that I would like to remind you that this individual case safety report was really the initial driver for developing the ISO IDMP standard. So this ICSR, there's a formal description and definition in the legislation. It relates to the format and content for the reporting of one or more several suspected adverse drug reactions. And when we talk about vaccine in specific, we actually talk about AEFIs, the adverse event following immunization. It relates to one or more medicinal products, and it can be a vaccine that occur in a single patient at a specific point in time. So we are really talking about an adverse event in one single patient. Currently, we have two formats of those ICSRs being exchanged. There's an old format that we refer to as ICH E2B-R2. And there's a new format that we use to exchange these ICSRs. And we refer to this as ICH E2B-R3. And this is the one based on the ISO ICSR standard. Of course, both formats have dedicated data elements to capture information on the drug. Things like as reported, the brand, dosage form, strength, a license number, substance, and the batch lot number. When it comes to the drug information, the main difference between the two formats is that the old format doesn't have dedicated data elements yet for the IDMP identifiers, like the MPID, the PHPID, and the substance ID. Whereas the new format, that we refer to as R3, does have these dedicated data elements. In the graph, you see a picture that I've received from an EMA colleague. And it shows the two formats being received currently by the EMA in the ultravigilance database. And here you can see that by now, still about 35% of the ICSRs is being exchanged in the old format. This is just for you to keep in mind when we talk about using IDMP identifiers in the ICSRs, that not every stakeholder is ready yet to use them in the ICSR. Next slide, please. Let's go back to this flow of ICSRs and focus on the brand and batch number, because this is really important. Does the reporter of the adverse event actually know exactly which vaccine, and then I'm talking about the brand and batch number, was given? If you press the button, you will see that this really depends on how the immunization program is organized. We have seen that vaccination registries are mandatory in the EU, but is it also captured at the level of the electronic health record? Or will the persons get a vaccination certificate? And if you press the button again, you will see a picture from my own personal vaccination certificate, showing that even if you have a certificate, it may not always mean that you know the details of the vaccine that you have been given. On top, you will see that I will only know which brand I've received. The second one, it's quite unspecified. It's just measles, mumps, rubella. And for the last one, at least here I know that the brand and the batch number. We do receive in our pharmacovigilance center currently still cases that are really unspecified, with just an influenza vaccine unspecified. And this is a shame because these are sort of lost for proper analysis. What also happens is that we receive cases with the wrong batch number because these little peel-off stickers are quite difficult to read. And we only find out that this batch number is wrong or doesn't exist because the ICSR, in the end, it has been sent to the pharmaceutical company and they contact us saying, this batch number is wrong, it doesn't exist. So there's a lot of work associated with getting those batch numbers right, if we have them at all. A good collaboration between the immunization program, the pharmaceutical company, and the pharmacovigilance center is really key here. Also, as Miriam said, having this little peel-off sticker at the primary package, at the vial, is not a given, especially not for the first rollout of the vaccines, because they have some labeling flexibilities for COVID vaccines in the EU. Next slide, please. Because we don't only collect all those ICSR reports, we also want to do analysis on them. And when we analyze them, we look, of course, for the overall pattern of those adverse events following immunization. But if we have the batch number as well, we will be able to see if these are different between the batches themselves, or if there perhaps are differences between the manufacturing side, because this can happen as well. We actually look at, I would say, three main pillars. Those reactions that are really related to the product, and we focus then on those events of special interest where Miriam's research has done a good job in collecting them for us and also finding the background incidences. We look at serious, non-serious cases, things like that. We know that for manufacturing and transport, there are a lot of checks in place already. But it does happen that we receive cases with adverse events that help us to identify a cold chain break or a falsification, or that there has been a problem in the storage as well. So this is a separate cluster to analyze. A third cluster relates to what we call immunization error-related reactions. And it could be an incident that the wrong needle has been used, but it could also be that at the vaccination facility, wrong instructions have been given. And then you have to intervene at another level. So this is what we do with all those cases. And in the next slide, of course, we don't only look at a specific vaccine, but we want to compare between vaccines. And as Miriam said, in the next slide, you'll see that there have been different platforms to develop the vaccines. And we also may want to do an analysis by comparing the viral vector vaccines, for example, with the protein-based vaccines. So this is all the things that we have to keep in mind and be prepared for. In an ideal world, we can do much more. In an ideal world, we can also look at the adjuvants, for example. Please press the button. Yeah, well, I am talking. We may also want to have a look at the products coming from the same manufacturing site if we have found an issue there. We may also want to have a look at similarities in starting materials, or if the product is based on a veterinary base material, from which species it's come from, for example, mice. I think you can press the button, or is it stuck? Yes, thank you. Thank you, Christian. And we can go to the next slide. Because one way the substance registration system can support this type of analysis is by grouping these kind of similarities, like common ingredients, common routes of synthesis, and I've put the manufacturing sites between brackets because it's currently not captured yet in EU SRS, but I think this would be a really nice feature to have. So for the next slide, to sum up, the collection and analysis of those ICSRs is one of the pillars for safety monitoring of the COVID vaccines. And as said before, brand and batch number are really key. And it's important that the reporters of those adverse events, the consumers, the healthcare professionals, know these details. The vaccines, the new vaccines, are based on new production technologies, and this requires a different type of safety monitoring, as each component may have its unique safety implications. And IDMP implementation can support the traceability up from manufacturing, supply chain, up to patient level, and provide the information to enhance the safety monitoring. And really my last slide is to make you aware that the EMA has a public meeting on COVID vaccines as well. And here you can find the program and the information. And thank you for your attention. And Christian, I give the floor back to you.
Thank you, Anya. Thank you for this very quiet way to present a complicated topic. And I'm sure you have been able to pass your message to the participants. Please note this hyperlink if you can. Otherwise, we will send you that after this meeting, because that's sometimes also difficult to capture, as is a batch number difficult to capture. So after Anya, we are going to visit Sweden, and the floor will be for Malin Flavad. She is explaining us what happens in her Uppsala Monitoring Center. Malin.
Thank you, Christian, and thank you for the opportunity to speak here. So I'm Malin Fladvad, and I'm working as a product portfolio manager at UMC, managing our dictionaries and also IDMP related projects. So I guess most of you know the UMC. We are operating the WHO program for international drug monitoring, which aims to ensure that the early signs of previously unknown medicine-related safety problems are identified, and very important that the information is also shared and acted upon. So the program gathers the individual case safety reports that we were speaking about early in a global data source, which facilitates a faster and more efficient detection of any problems that could occur. So each member country in the program, they collect the adverse events reports from their populations through their pharmacovigilance programs. And then they analyze all the data there in their country, and they use different methods for collection. Around 90 of the 150 members, they use the UMC tool Vigiflow for managing the ICSR data. And the data are then shared with the global database. And from there, we have a tool available for all the countries, which is called Vigilize, where they can look at the global data and compare it to their own national data and use this to detect issues. There is also a tool for presenting results on a summary and aggregated level, which is called VigiAxis, where all of you can access this global database. So in all of these systems, so from the reporter to the ICSR management and to the analysis of the data, of course, the drug needs to be identified in each of these stages. And I would now highlight to you some of the specific examples of problems that could be related to the identification. So can we go to the next slide, please? Thank you. So what kind of issues can we expect? Well, here we have an example of identification of a particular brand and age group related adverse event. So in 2010, administration of influenza vaccine in Australia resulted in an estimated rate of 3.3 febrile seizures per 1,000 doses administered. And this was among children below the age of 5. And the risk of an influenza vaccine associated seizure like this, in this case, it was highly elevated compared to the previous years, and it was found to be associated with the vaccine formulation of one manufacturer of the influenza vaccine that was distributed that year, the Fluvax vaccine from the CSL Biotherapies. So a study of the events found that there were excessive viral components in the vaccine, and it was suggested that the combination of a new influenza strain, the increased complexes of viral RNA and lipids in the vaccine, together with the sensitivity in some children for these kind of events, caused an elevated inflammatory response. So here you can see how complex it can be with the vaccine, and also that the analysis needs to be specific also regarding, for example, age groups. So then if we go to the next slide, we can look into another issue, and that's the risk of the naming confusion. And a very good example to illustrate is for the various age-dependent combination vaccines that targets diphtheria, tetanus, and pertussis, and polio. And then there are additional variants with components also for influenza and the hexavalent vaccine also targeting the hepatitis B. So what leads to confusion in this large number of vaccines is that there are so many various combinations. And there is also differences between the recommended ages for the individual components of these vaccines. There is also confusing labeling sometime of the package. You can see that on this picture that sometimes they are actually quite similar. There's also in many cases a large number of words related to the product name, and to squeeze that information on a very small space available on a single dose vial, that's almost impossible to read in the end, and similar for a batch number. There's also sometimes large similarities between the generic and the brand names, and also in the abbreviations used for the vaccines. You can see here you had the, you have to really be careful to remember these different abbreviations and not to confuse them with each other. So for the COVID-19 vaccines, we hope that we will get a really proper labeling and also the uses of barcodes to avoid these kind of problems. So if we go to the next slide, as been pointing out earlier here by Miriam, there are new vaccines that are coming now for COVID, and the platforms are not really explored that much. So another issue that we are hoping to be able to monitor on a global scale is potentially related to the platforms. And we think that that's very important. So then, and in this case, it's very good to have specific and detailed information about the substance itself to be able to actually pin down what the problem could be. So if we go to the next slide, I would talk a little bit about the actions that UMC are taking now for COVID. So first, one important task is of course to enable the program members to perform the signal detection and addressing both the local, national and global needs. So right now we are implementing an IFI specific entry form. The ICSR management system that we are using normally supports the processing of spontaneous ADR reports, and the structures and processes for the AFI detection is slightly different. So in the management system, the national authority will first make their own assessment to identify if the problem is regional and possibly related to, for example, cold chain issues. So there has, however, been issues with the IFI form itself that have been highlighted. For example, we have had discussion regarding the information about the manufacturer versus the market authorization holder. So normally for spontaneous reporting, we report on the market authorization holder, but for vaccines, the manufacturer information is sometimes more easy to get hold of. And this is also something that could lead to confusion. We are also improving the analysis functionality to ensure that the vaccine substance name structure enable fast and efficient analysis, for example, for the platform issues. So similar to the proposed structure for medicinal products in IDMP, the Program for International Drug Monitoring are using the WHO drug dictionaries, and there the substance information is linked to each brand name. So with the lack of a current global substance identifier, it's very important to have good substance names that are short and still makes it easy to identify the vaccine and the platform itself. And we are also developing complementing drug groupings to ensure that the analysis should be as easy as possible for the program members. So then if we go to the next slide, we of course also have the work to perform the signal detection and analysis for global adverse events reporting patterns. And we are currently reviewing the medicines and the medicine groups of interest for COVID, like for example, remdesivir, glucocorticoids, and the heparin class medicines, and many more. So far, I would say that the ADR patterns are mainly consistent with those that have been described in earlier reports, and mostly within the labeling. So these kind of information is then published, so the regulatory authorities can get this information directly from the Visualize UMC tool, and others can view it in the WHO regulatory updates. So the next focus for these analysis will of course be the COVID vaccines. So then if we look into IDMP more closely, UMC are also working on a pilot on the PHPID within the UNICOM project, and we have completed the first set of PHPID generation. So regarding PHPIDs for COVID vaccines, UMC is prepared to issue them from a technical perspective based on the work with the pilot and the information available about the dose, form, and strength of the vaccines. We could also link to appropriate other information, for example, substance IDs available from EMA and also FDA, and to GTIN if that's available. There has, however, not been any agreement on a formal global substance identifier, which is necessary for a global PHPID. But the solution now is that we will create unique substance IDs that meets the purpose and the quality according to the ISO standard for unique identification. So however, regardless of UMC's preparedness and the technical ability to generate these IDs, there are many questions that remains. Are we going to be able to use this for this pandemic? Who is going to use it and how with the operation work. So I think we still have a lot of discussions that should be ongoing, and we need to get the proper people involved. As Anja, if we go to the next slide, as Anja previously described, the work on vaccine substances is part of the EU SRS implementation project in Europe. And we are now working on a more transatlantic and potentially a global approach for vaccine cleansing, together with the EU SRS team and colleagues from the FDA. And the idea is to work on the vaccine substances in a shared GSRS instance that is hosted by UMC, and where the EU and US colleagues can jointly agree on the vaccine data and also get important input from vaccine experts in the industry. And when the data is cleansed and agreed on, it can then be pushed out to the local SRS systems, to the EU SRS and the US SRS system. So we think that this approach has several benefits. For example, it would increase the resources with expertise in the area of vaccines, and also that the involved parties will have a unified description of the vaccines. And then, of course, the industry involvement is very welcome, since they have the knowledge in-house on the vaccines, which will reduce the time to search for very specific information. So if we go to the next slide, I just want to summarize, and in principle, I will say the same thing as Miriam and Anja said, that without the IDMP implementation, we will have to rely on the brand name information, and in some cases, substance information where it's available, and together with the batch numbers. But I think we should really aim for implementation of IDMP with the IDs that are available there for the medicinal products, the PHP ID and the substance ID, and especially together with the global substance repository, that would really enhance the possibilities for identification and analysis of safety issues, especially for COVID-19. So that was all from me. Back to you, Christian.
Thank you, Marine. I kept Miriam's last slide with a question because I thought the illustration is just fine to make the transition from the free speeches we had now to the questions, and we will keep that a moment on the screen. The speaker and panelists can open their camera now, and I'm going to look a little bit what we have for questions. I noticed for a while that Ursula Chorn raised her hands, and maybe I give her the permission to talk.
Oh, thank you, Christian, for that. Yes, thank you. First, very interesting, your presentation, and as part of UNICOM, I'm very proud that we are really working on these challenging questions, and hopefully we will succeed in being in time. My question was about the Access project, but I've got already the answer in my chat. Thank you for that. That I may look at EMA for this Access project to know how it is working in direction of product identifying and batch identifier, to know if there is special work already done, which we can perhaps take in our pilot product list for UNICOM.
Ursula, I can quickly answer, yeah? But nothing around product identification has been done as part of the Access project. The Access project has written protocols where, if, based on the spontaneous reporting databases or the ICSR that Anja and Malin were talking about, if there is a signal that you could quickly run studies that would look, how big is the problem, and also whether there is, to quantify the risk, but they haven't worked on the identification. So that's, I think, where we are all coming together, that there is a clearly recognized need, both in ICSR for the spontaneous reporting, as well as from any type of post-introduction study, that there is an that the identity of the product is clearly identified, and so there is I think it would be great to have the IDMP, but that hasn't been fixed yet.
Thank you, Miriam. Does someone want to add to this answer? That doesn't look. I see Carl Sotman has raised a question to Malin. He is asking, the activity on EU SRS for vaccine will be limited to new vaccine, or also to old ones that are still included in the vaccination card? Malin.
Yes, thank you, Carl. No, it's not limited to new vaccines. We will actually start off with the so-called old vaccines. And then as long, we are hoping as for when the COVID vaccine comes out, that we will get them registered as well. So this is a pilot, I would say, but we are really hoping that this will be a good and a start of a possible global collaboration.
Thank you, Malin. I see another question here. Is there not an added dimension not currently discussed, namely the implication of COVID-19 vaccine within the framework of the Falsified Medicine Directive? Meaning, are these products going to be sterilized? The answer is yes, they are going to be sterilized. It has been part of a memorandum of understanding between the European Commission and the member states. And that is published on the, I think, on the Commission website. I couldn't give you the link, but I can send that afterwards. And the special aspect of it is that each of these vaccine will have one single GTIN for all the Europe. And okay, that will be sterilized. So for the Falsified Medicine Directive implementation, I don't know exactly how it will work regarding the target countries and so on. Do we have another question? Then I have another one because of course we are here in Europe and looking for ourselves first. It's a bit selfish, but actually the immunization programs will be more global. So I would ask Malin if she can expect to have feedback, in the sense of reports coming from lower or medium income countries with the vaccine which are going to be distributed there.
Yes.
How will that work?
Yeah, so I think for many of these countries, they are actually using the ICSR management system that UMC provide. So what we are hoping now, when we are implementing better functionality for vaccine reporting, that they could use this potentially if they want to report, if they don't have other systems, which is even more appropriate for vaccines. And we are also collaborating with the vaccine programs in many countries, and are now trying to improve also the lag time for reporting, because that's one of the major problems, that the lag time for sending the reports to the global instance is sometimes very long, and then in this case it's not really that useful. So that's some of the parts that we are working on.
Okay, thank you. I see another question, which is for all of you, I suppose. The question is, there are already some experience with mRNA to prevent infectious disease in animals. Any use or leverage to anticipate pharmacovigilance for humans? Who wants to answer that?
Anja, do you want to? It seems more regulatory, but so we know some of them have been utilized. It's always difficult to translate directly to humans. So the absence of anything happening doesn't guarantee that there would not be nothing in humans. So I guess that's the situation where we are. Anja, do you want to add?
No. And to be honest, I'm not that's an expert that I know about this veterinary pharmacovigilance translation to human. So I would have to check with my veterinary colleagues as well what they observed and how we can learn from that. So thank you for raising this.
Any other reaction, Malin, on your end?
No, I'm not a veterinary expert at all. So.
Good, thank you. I see the question which I mentioned before about the serialization of the vaccine for COVID-19. That would be answered as well by Robert van der Stichele. Robert.
Serialization is the basis of the protection against falsified medicines, which I think will be important, but we can be sure that falsification will occur also in this new and very, very stringent area of COVID vaccines. So if companies have been hesitating to do this because producing a serialization number, so an individual number for each box, can be time consuming and would hinder the race to the market, especially in the beginning. So that's, I think, one element to be considered. But it is absolutely essential that a serial number is at least at the secondary packaging, the boxes. How these boxes will be for COVID vaccines, they will not be the traditional cartons of medicinal products. They will more be bigger boxes, probably like pizza boxes, sometimes with a cooling system. But anyway, a serialization number on these boxes, on the secondary packages, seems essential. Once you talk about serialization numbers, batch numbers, and a GTIN, these are already three complex numbers, it would be very handy that these three numbers are translated in one rectangular, two-dimensional barcode, so-called data matrix. And, but then it means also that you have to equip the people who will actually do the vaccination, and in the beginning it will be done in specialized vaccination centers with specialized vaccinators. They need to be equipped to read these barcodes, transform and in the same time for reading, transfer the information to their system, to the vaccination register, to the GP record, and eventually also produce a certificate for the patient. So I think if we really want to implement the protection for falsification and the batch number and the GTIN number, the product identification, we will need to have these barcodes on the secondary package and vaccinators who are equipped to read them and systems that can process these numbers. And that will make it very easy. It's probably difficult to realize this immediately in all countries. At least developed countries should think in that direction. Low and middle income countries are also preparing for this. In fact, some of the pilot products from some manufacturers for developing countries have pioneered this new approach, and I think it would be COVID-19 could be an occasion to pioneer a whole new system of vaccine identification, also for the future and for other vaccines.
Thank you, Robert. I just see an in-between question about using radio frequency at a different level of packages for the same purpose. But this is not really on the roadmap at these days. When you think that we are still struggling about using an optical data carrier such as data matrix and capture that, radio frequency will be a bridge in all what we are doing now, and between now and Christmas, it is very unlikely RFID will be used in that space. And adds to that it happens that some of the vaccines are liquid to be injected. RFID is not really appropriate for liquid identification anyway.
There are also QR codes.
No, data matrix.
Yeah, but yeah, I know, but you have RFID, you have the two-dimensional data matrix, and you have the QR codes. QR codes are just a graphical representation of a URL. It points to some website where information can be read. It can be useful in the case of labeling, but I think it's not the right technology for what we're talking here, identification of the product, batch number, and serial number.
Thank you. We have two questions. One is raised for Anya primarily. We have the question, has any study been done on how the ICSR reports are embedded in European member states in terms of structure, content, and especially in those countries where electronic health records are subject to official certification? Anya, do you have a, yeah?
Thank you for that question. Yeah, every member state is by law required to have systems in place to collect ICSR reports. And actually in the UNICOM project as part of task 8.4, next year one of the activities will start to do this kind of study. What has been done already in the countries, and what you see in some countries that there are at national level some infrastructures in place, but yeah, or targeting a specific group, like in the UK, they have systems in place for ICSR reporting from GP systems. But when I look at the Netherlands, for example, it's quite regional, really local for some particular hospital systems. So I think the landscape is very diverse. And in task 8.4, we are really looking forward to start this work as well.
Thank you, Anya. We have an interesting question also. All the questions are interesting, actually. An interesting question for Miriam. Could you share your thoughts regarding how the effect of migration could impact the success of national immunization strategies in Europe? And that person is specifying, we speak here about intra-European migration, but also out of Europe, but mostly intra-European. Miriam, some thoughts about that? I notice your camera is off now. I will put mine on. Voyons. Okay, Miriam is on mute, so we may skip that question for now and ask her right after. We are coming back to basics. These are the ones I understand the best also. A question is, can a data matrix be read by a smartphone? The answer is yes. Sometimes it is not native in the smartphone itself, but you can have an app for that, and there are apps enabling a lot of things. The process to have a native app in every new smartphone to be able to read such a data matrix with the GS1 data structure is ongoing. Another question, because it's related, I think, is there any provision to require that all systems would be able to transmit this data properly? Data is adverse event reporting. Even if you capture all those information, if there is only one system that does not transmit them properly, then we lose traceability. Who wants to speak about that? And I can perhaps say that the question comes from IHE, an IHE representative into the organization which is running connectathons to test that such communication works efficiently. Robert?
In an ideal world, it would be best that all relevant organizations who need the information have the information. I do not agree that if one system doesn't work, then the traceability is lost. I think it's good to have the data in a few places. So if there are glitches in one system, you can always rely on another one.
Well, it depends a bit what one means with that.
Yeah.
It depends. I see Miriam is still not back, so I will repeat the question regarding migration when she is here. Voila, she's arriving. Fabulous. Miriam, we had a question for you. Could you share your thoughts regarding how the effect of migration could impact the success of national immunization strategies in Europe? And that person is specifying migration which can be intra-European, also extra, but mostly intra-European, so movement of people inside the Union. Do you have a thought on that?
So migration, I guess I would need a bit more of clarification, because migration can have multiple impacts. So first of all, if somebody is being vaccinated in one country and would move to another one and would have their second dose there, it would be important that it can actually be recognized what was the first dose of vaccine. So, you know, having the ability to trace back what has been done is important. Secondly, I think migration in terms of like traveling across Europe, now, I mean, it's largely forbidden if you are in a red zone country. We may anticipate that once you're vaccinated, you may have more opportunities to do this. And so you would need to have a certificate that shows that you have been vaccinated. Data migration between Europe, I don't think that data will travel so quickly, and that is not needed as long as you can access data in a national country. So if you have an ability to understand that somebody has been vaccinated, but you can keep the original data in a country, you don't need to shift around data in Europe. You can actually, if you have a pointer towards that data, and the result of it would be important. And that is the way that most of these studies are being done. So yes, so far, I hope I answered the question.
Oh, certainly, yes. Thank you very much, Miriam. Robert van der Stichele wishes to add some thoughts on that.
Maybe for the migrants in Europe, the whole question of monitoring will be an extra challenge, and we will need a specific monitoring plan for how this will be monitored in the migrants, which are not always registered in the e-health systems of the countries.
Yeah, it is obviously a challenge. Yes. That may also raise our attention to the immunization certificate, which is also in the thoughts, which would be partly certainly solving this kind of issues. But I suppose that will also not be on the Christmas tree. I have a new question. In addition to monitoring adverse events, will manufacturers monitor efficacy of vaccine by making examination of the case of COVID-19 diagnosis among patients who were vaccinated? Well, it is a question to the manufacturer somehow, but it is a general question as well. Who wants to answer that?
Yeah, I can answer that. So in general, when the vaccines are licensed, there will be a risk management plan which will detail what are known, what is known, what is unknown. And there is an obligation, I think, upon for every drug that the benefit-risk ratio is being monitored throughout the life cycle. So if there would be doubts, so it's actually upon licensure, there will be studies agreed or not agreed. I mean, it will be discussed then what is known and what needs to be understood. But clearly this is an option that it can be done. And I think one of the key concerns that people are expressing is it's the potential possibility of a vaccine-enhanced disease. So that if you have been vaccinated, that you could have a more serious COVID. We don't know whether it can happen, but it was seen with MERS, for example. So there is this potential, although it hasn't been seen yet in the clinical trials, but that's something that needs to be monitored. So it's almost like effectiveness towards COVID-19 needs to be monitored to be able to even spot like this potential side effect, which is enhanced disease. So it comes very close into, whether that's efficacy or safety monitoring, but it needs to be monitored.
Thank you. Well, we have somebody challenging the question itself about this migration, which I was reading twice before. I think the question should be a bit framed and has nothing to do with illegal people, but it has something to do with some people who are moving inside Europe and have their immunization, say, in part somewhere and in the second half elsewhere. And that has been answered, I think, very properly. So we need a system able to communicate from one member state to another one, so that the immunization acts can be accessed, and can be updated. And no, that was not a question we had in the past to answer that last comment in it. I think Karl Strootman wants to say something still in this space. I will give him the microphone now. You can unmute yourself.
Good afternoon. It is not Carl, it's Marcello.
Ah, you changed the accent.
No, it was like before. Sorry, no worries. It's an honor to be confused with Carl. No, related to this point of migration, there are two activities ongoing. One that is a medium long term. There is an ongoing project for the creation of the European vaccination card that has a goal of, and it is a DigiSante project, has a goal of transferring all previous paper vaccination card into something electronic. That's why I was asking about the data cleansing for vaccine. But related to the migration, there is an urgent activity started by the EF network, meaning the board of the Ministry of Health at European level, to have the vaccination certificate up and running with the first vaccine. So now it is really impossible to have it in December, but it is planned to have a service that will allow a certification of the performed vaccination for COVID mainly, for all European citizens as soon as they receive the vaccination. So even the concept of having the first amount and the second amount should be tracked. And there is a request for allowing traveling cross-border. And as I said, it is on the top of the activities from the EF network that has been assigned to the EF network subgroup on technical. And there is a team that has already produced indication and a sort of four possible alternatives on which the EF network should take a decision next Wednesday. So not under the Christmas tree, but maybe for the Epiphany, as how to continue. Thank you.
Thank you, Mr. Carl Marcello. But just to be more specific in your question, or your answer, I would say, we had some speaker highlighting before the need not only to know which is the vaccine used, but which is the batch. And can you just say, if you, according to your information, the batch will be included in this?
Yes, in the vaccination card, it is foreseen to have the batch, absolutely. When available, of course. In this vaccination app is much more related to the concept of contact tracing on one side and having this. But since the people that are working on that are expert on vaccine, for sure the concept of having also in the repository, in the record, let's call it generally speaking record, also the batch is a point to be highlighted. And just to say that this is a very urgent ongoing activity. Yes, I can't give the document now because it's very restricted, but there is already a document.
Excellent, thank you. Do we have another question? I see none at the moment in our Q&A box. If I would have missed a question, that person may repeat the question because I see only the last questions on the box. I see nothing coming. I am going to close this session. Normally we have that questionnaire which I forgot to use, of course. Well, where is it? Oh yeah. No. Oh, it has disappeared from my system. What is it? That is a shame, and it's really not good. Then we will skip the questions. I'm sorry for that. It's not the time to do it now. We have our next community of expertise on the Friday, the 22nd of January. I would like you to take the opportunity to bookmark that in your diaries. Same time as today, same procedure to register. It will be dedicated on pharmaceutical dose form, PDF. We are having a couple of presenters for that. It is a representative of EDQM, Christopher Jarvis. We will have Robert van der Stichele, who is also here on the panel. He will be a presenter. And a special guest from the US, Ron Fitzmartin from the US FDA, and representing, he's not the only one, but representing the US in the ISO working group 6, where we discuss all these things. We will have a panelist also in January. That will be our Swedish guest today, Malin Fladvat. She will then be panelist to contribute to the discussion between all that. So please bookmark 22nd of January. And now I have probably a closing remark, a closing question from Karl Strootman. This time it's probably the right one. You can open your microphone, Karl. That should work. Alors, voyons. Where is your microphone? If you cannot activate your microphone, that will be...
Can we bring up the figure from the manuscript to answer the question of Karl?
I haven't seen the question of Karl yet. Wait a minute.
What is a serial number, a batch number, a lot number?
Can you hear me now?
Yes.
Yes.
Okay, yes. I lost connectivity for a while. I just see everywhere you talk about serial number, batch number, lot number, and I saw this barcode picture, and I must say I'm getting a little bit lost. Yes, exactly this picture I meant.
Yeah, but can you bring up the other picture?
Yes, it's much better, but wait a minute, who is it? Where is that?
It was in Anja's presentation.
Yes, I'm searching. I have to go slowly because
yeah,
my
you're getting there. You're getting there. Next one. Yeah, that one.
Yes. Maybe if I make it larger like this. Voilà. Robert, you want to explain?
Yeah, this is the illustration of the upcoming article commentary in the journal Vaccine, where many of the 4 of the 5 authors are from UNICOM. And maybe we can start with the smaller picture. So we have the overview. Yeah. So vaccines come in primary packages, which is the vial or the syringe. Then the secondary packages, which is a box that is usually used to distribute it from community pharmacies to patients or to physicians. Bigger box in hospital distribution, and then pallets for wholesalers. We are interested in the primary and the secondary. And the idea is to have, if you look carefully, and now we can maybe make it greater, the 2 data matrices. The first one on the primary package, so on the vial, has the GTIN number, which is quite a long number, as you can see, GTIN. The expiry date, which in the case of COVID vaccines is for the moment not at all known. It's a big question mark how long these vaccines will last after production. There will be a date, but it will be not so meaningful at first. We need more time to be able to determine reliable expiry dates. And then there's the lot number. You don't see the serial number here because that would be complicated to put it on every single vial. If you look at the secondary package, you have again this data matrix, but this time with 4 numbers. And the 4th number is indeed the serial number. And all these 4 numbers are captured in one little rectangle data matrix, which can be read by a dedicated scanner or by a hand phone, and then transferred to whatever system, to the certificate, to the national register, to the record, to the health record of the GP. And that is so crucially, so basically crucial that at least on the secondary package of the vaccines that are rolled out now, this information is available. We already know that for the whole of Europe, the GTIN for the first vaccine of Pfizer will be the same throughout Europe. But we will have a batch number, should be on there, and there should also be a serial number. All these numbers sketched in one rectangular, 2-dimensional data matrix barcode.
So is batch equal to lot number?
Yes, batch and lot number is the same, yeah.
Okay, that was confusing me.
A synonym, yeah.
Thank you, Robert, to have answered that question. We have another Robert Stegway, my chair of now Work Package 1. He wants to answer comment the requirements for Marcello Melgar before. Robert.
There was a question to Marcello, and I just wanted to make sure that everybody heard it and that Marcello could answer if he wanted to. Whether the requirements that you mentioned on the vaccination, what was it? Certificate, whether that was actually, if those requirements are publicly available, and if you could point us to them. Marcello, are you still there?
No, I was muted and I'm muted. It is an ongoing activity, so I prefer that first are adopted and considered by the health network before sharing it. So it's better that we keep. It was just a positive signal in that we are working on it, several people, and we know that even worldwide people are heavily working on this.
Yeah.
We are also looking to the worldwide activities. And but in order to disclose something, it is better to wait for any official decision.
The IATA is working on this. WHO
is working on this, but on a purpose of admitting people on the airplane And the way in which IATA is working is not fully in line with what healthcare institution or Ministry of Health are really willing to.
Okay.
So yes, we know that's the situation.
And WHO and Estonia also have a project.
Estonia was considered. WHO is the queen. I don't know if a queen or a king, but he's our queen or queen.
Okay.
I don't know the sex anyway. I prefer to say the queen. I prefer ladies, you know.
Well, it cannot be the Father Christmas because you said if something happens, it will be at Epiphany, which is after Christmas. I think we are at the end of this committee of expertise. We have lost a couple of participants already. It's actually exactly 4:30 now in Europe. I would like to thank the panelists and the presenter, and I apologize that some person want to say something about another question, but we have to close it now because it is also a question of politeness. When we say 90 minutes, we should stand to that. As I said, the next committee of expertise in a few weeks from now. When you have questions, please ask them in the flow of the presentations. That allows us also to structure a little bit when we want to group questions for a combined response. It's always better. I hope this session was useful to all of you. Maybe we will be in the same virtual room in a week with the European Medicine Agency. But certainly we will end the year mostly on the same day or an hour. In a few weeks from now, I hope you will have a safe period of time. I look forward to see you next year, still as dynamic as now. Thanks a lot to you all, and Happy Christmas.
Bye-bye. Bye-bye.